On slide 21, what we're looking at is a cross-section through a testes. And we even had, on the model, the male reproductive model, you had a slice through the testes that you could see all of this. Now, there are some portions of it that are labeled. And so you can see to the right, you have the seminiferous tubules. And they go through this something called the ready testes, which is this, sort of where the seminal tubules combine together before they get to the epididymis. And basically what happens in the seminal tubules, this is where the sperm cells go through a process called spermatogenesis. So when we talked about eggs maturing to become an ovum, we had called that oogenesis. Here, when we talk about the conversion of a regular primordial sex cell, which is a regular cell, into that highly modified cell we call a sperm cell, this process is called spermatogenesis. And so spermatogenesis happens in the seminiferous tubules. Now, and we'll talk about that on the next slide, but after they mature, where do they go? Well, they go and hang out in the epididymis, and they can hang out there for, I think it's for 60 days, something like that. For a long period of time, that's where they're going to go. So they're going to be made in the seminiferous tubules and stored in the epididymis. And then if you look carefully at that epididymis, you see what? You see it's connected to the vas deferens. Let's see what term we're using for ductus deferens is the other version of the vas deferens. All right, so let's look at one of those seminiferous tubules in cross-section. So just one that's been cut off, so you're like looking down the end of the pipe, so to speak. And we're on slide 22. And we have some interesting structures in here. You can see some really big cells that are along the lining or right along the interior surface of that seminiferous tubule, and they are called Sertoli cells. And they're kind of like the mama. they help protect, they nourish and regulate. They are the cell that is going to be helping out those developing sperm cells. So what did we said? We said that, and if we look at our picture, you can see the spermatogonia, and that these are the cells that become the sperm cells. And you can see on either side of the Sertoli cells, and if you look at them, you can see they look just like a regular cell. Well, what's going to happen is those spermatogonia are going to lose their cytoplasm, and they're going to become essentially just a nucleus with an enzyme on the tip of it, a midpiece, and a tail. And they're going to be, and so they can't do anything for themselves. So the Sertoli cell is the one that is responsible for taking care of them prior to them going off to the epididymis. Another set of cells are there, and you can see they're called interstitial cells. And so if you think of the tube and then the spaces between the seminiferous tubules, this is where the interstitial cells are. And the interstitial cells are going to be the cells that are going to secrete testosterone. We said the testes, they're basically what they do is they produce sperm cells and secrete testosterone. Interstitial cells are the ones that do that. All right, moving right along. Slide number 23. We see an interesting diagram, which is showing, I think, I can't see it very well on mine here, is showing from the bottom. Okay, so yeah, to orient yourself, you have the spermatogonia at the bottom of the slide, and then they're maturing as they move up until you end up getting the mature sperm cells. And so this is, you can see they start out with looking just like a regular cell, and they become modified into spermatogonia. Just looking at my slides, basically, of course, until sexual maturity, you're not doing this. Then at puberty, basically you start to produce the sperm cells and secrete testosterone, and that's why little boy sopranos become baritones, right? Their voice deepens and they have to start shaving and all of that. We had said we have this special form of cell division where you reduce the number of chromosomes by 1/2, so that when the sperm cell unites with the oocyte or the ovum, you have one set of chromosomes from mom and one set of chromosomes from dad. And this is dad. All right. So here again, slide 24, it's showing this process of spermatogenesis. That's what we call this, the conversion of the spermatogonia into the sperm cell itself. And so if you look at the diagram of that sperm cell, you can see it has a flagellum. It has a midpiece. Now the midpiece has a whole lot of mitochondria. What do my mitochondria do? What? It's the powerhouse of the cell. So in that semen, we are going to have nutrients so that the sperm cell can swim. What does the flagellum do? It's like a little propeller that propels the sperm cell along. The mitochondria is there to take the nutrients that are in the environment, the semen, and power that little bugger, power the propeller. And then we have the nucleus, which is the head. And we had said this is where we have the single set of chromosomes. And we have one more thing there. On the tippy top of the sperm cell, we have something called the acrosome. This is a packet of enzymes. We said, what does that sperm cell do? If it's the lucky one that gets to the ovum first, the acrosome is what allows it to penetrate the egg and take part in the blessed event, fertilization, right? So it's the acrosome is enzymes for penetration into the ovum. Okay. Yeah, and so the slide 25 saying all that same stuff, right? Okay, so 26. We have this seminal fluid, right? I'm trying to see. We have the bulbourethral gland. We had said that it's really more about getting the urethra ready. But then we're going to be talking about the semen itself. I'm going to go on and stop right here, and we'll pick up our discussion right here in just a minute.